Morning all. It is an honor for me to moderate the two distinguished guests. I would like to call upon Professor Ann Kelso. She will present a lot of her expertise, experiences, and the current research and the current and the future, what we have to see on influenza. So her topic will be on surveillance, evolutions, and prospects for control of influenza viruses in our region. So, Anne, time is yours.
Thank you very much for that warm introduction, and special thanks to Professor Tegu for organising and inviting us to speak here. Thank you. It's really a great pleasure and an honour, and I'm very grateful to UGM, to the Australian Embassy, to the Indonesian Academy of Science and the Australian Academy of Science for organising this session. And it's really a wonderful opportunity for me to meet you. I've been to Jakarta several times, but this is my first visit to Yogyakarta, and so my main disappointment is that there is no time to have a look around and see your beautiful city. But I know that it has many interesting things for tourists, so I promise I'm going to come back as a tourist soon. So it is my great honour to talk to you about influenza, and I hope that this will be clear and interesting. And I don't know whether you want to do this, but if anything is unclear, I'd really like you to look very worried so that I have an idea when to slow down and when to explain further. And so just to start by a little bit of introduction to what I want to talk about. Now, influenza is, of course, it's caused by a family of viruses, influenza viruses, but it presents three quite different challenges to humanity. Seasonal influenza, pandemic human influenza, and avian influenza. Now, seasonal influenza happens every year all around the world, and it causes something like 250,000 to 500,000 deaths worldwide each year. Not a big one like HIV or TB or malaria, but nevertheless, year on year, through the years, the decades, the centuries, influenza causes many, many deaths. Pandemic influenza only occurs from time to time. It's quite unpredictable, and I'll talk more about that. But it can cause extremely severe illness. The 2009 pandemic is estimated to have caused about 300,000 deaths in its first year worldwide. So that's a small pandemic, a mild one. But the 1918-1919 influenza pandemic is estimated to have killed 50 million people in its first year. That was something like 2.5% of the whole world population at that time. So that is a great concern for public health authorities. And then avian influenza, with which I'm sure you're also familiar, is a very severe concern for poultry farmers. It doesn't cause so many human deaths, but it's very worrying because of the potential for avian influenza viruses to become pandemic. The H5N1 virus, you may be familiar with, has killed about 390 people as far as since it re-emerged in 2003. And the new H7N9 bird flu virus, which emerged in China just a year ago, a bit over a year ago, is thought to have killed about 120 people so far. So I'm going to talk about these topics. I'm going to say more about seasonal, pandemic, and zoonotic influenza viruses. I'm going to talk about global influenza surveillance done by the WHO and our own involvement in that work. I want to talk... Can you hear me? Yes. A little bit high? So my arm will get tired. I'll know when to stop when my arm can't hold up the microphone anymore. I want to talk about zoonotic influenza viruses and the particular threats they present today. Now, if there's time, and there may not be, I want to tell you about two current research projects, and then finally I want to make a few remarks about the prospects for control of influenza. So just to say more now about these viruses and to explain the difference between seasonal and pandemic viruses and where they come from. Seasonal viruses are human-adapted influenza viruses. There are three types, A, B, and C, and these are circulating around the world all the time. They never disappear entirely. And you can see for these Australian data that they are highly seasonal in temperate climates, and most Australians live in a temperate climate. The spike happens every winter, which for us is between about May, June, September. It was very high in 2009 when we had a new pandemic virus, but you can see it just varies a lot from year to year. Sometimes there's a bit of it. So we see in temperate climates this very spiky occurrence of influenza. In the tropics, there is also circulation of influenza viruses. It tends to be more throughout the year and varies a lot from country to country. Types A and B are the influenza viruses we worry about. Type C is not very important. And at the moment, around the whole world, there are two types, A and B, and two subtypes of A, which are called H1N1 PDM09 because they came from the pandemic virus of 2009, and A H3N2. And then there are two lineages of influenza B viruses that are constantly co-circulating in the world. Some years there are more of the Victoria type, and sometimes there are more of the Yamagata type. At the moment there are more of the Yamagata type circulating in the world. For countries which have seasonal influenza vaccines, normally they're producing a trivalent vaccine with three components, one PDM09 virus, one H3N2 virus, and then one of the B viruses, either Victoria or Yamagata, depending on which one is predominant in the world. Some countries are now starting to use quadrivalent vaccines, which have four components, and they have both a Victoria and a Yamagata virus. So in Australia, I've said that most people live in a temperate climate, and that's because although we have the Tropic of Capricorn running through the middle of our country, so quite a bit of our country is subtropical to tropical, most people live in the southeast and are therefore in a temperate climate, and therefore on average you see a temperate pattern of influenza circulation. But it varies a lot from region to region in Australia, and so you see up here in the Northern Territory, which is actually very close to Indonesia, we see often two cycles of influenza each year. So this is typical of many tropical regions and countries. In Queensland, which is a mixture, we see kind of a mixed pattern, and then further down south, like Victoria, where we live, it's much more of a temperate pattern. So it varies a lot in different parts of the country and different parts of the world. Now, there have been a few studies about seasonal influenza virus circulation in Indonesia. I found three papers the other day which show very nice studies of what normally happens here. And I've used this figure from one of those papers published in 2012. And it shows, this is averaging data for several years in the 2000s, shows that you have influenza circulating throughout the year in Indonesia, but it's highest in the wet season. And so it never goes away, and I guess right now there's a significant risk of catching seasonal influenza in Indonesia, but it's higher in December, January, February. So you clearly have something that's a tropical pattern that's seen in many countries. Yeah, yes, of course.
Because we are doing a live streaming, so it is actually very important for you to either to put the mic closer to you, or you may sit down and then you can see it from here, because then you can use this, might be better. Because this is a live streaming and we also record it. Is it okay?
The problem is mainly the microphone?
Yes.
All right. Shall I see how I go? Because it's nice to stand.
Yeah,
but
exactly. Yeah, maybe a bit
it's
closer.
quite hard to do that, isn't it?
Is
But
it
yes,
already?
with pleasure.
Yeah,
I will try to do it.
Yeah?
And please let me know if it's not working very well.
Kalau
I
sudah mulai turun
can
lagi,
tell it makes a difference.
make a sign. So
Okay.
I asked him to make a sign if it goes kind of soft.
Yeah.
Yeah.
Yes.
So
Okay.
sorry.
No, that's fine. I certainly want everybody to be able to hear it and for the recording to go well. Now, pandemic influenza is different. It's caused by influenza A viruses, and these are new viruses which aren't previously adapted to humans, and they emerge irregularly. And this shows an analysis of historical data that suggests, we can't be sure, but suggests that there have been at least 13 pandemics over the last 500 years. And you can see that they are completely irregular. So there's a very long gap of 150 years there, but sometimes a gap of only about 10 years, as in between 57 and 68. So you cannot predict when the next pandemic will happen. You cannot say we are due for a pandemic because pandemics don't follow a clock. Now, when a pandemic virus emerges, very few people have immunity to that new virus, and so the virus might infect more than half the world's population in the first year or so that it circulates. Those infections may be out of season in temperate climates, but eventually that virus becomes adapted to humans and it becomes the seasonal virus. And so our current viruses, the H3N2, which emerged in 1968 as a pandemic and continues to circulate as a seasonal virus, and the H1N1 PDM09, which emerged in 2009. To understand how pandemic viruses arise, we need to understand the ecology of these viruses. And it turns out, perhaps many of you know this, that the natural host of influenza A viruses is water birds, particularly ducks, shorebirds, birds that wade in the water or dabble on the surface of the water. But these viruses actually don't normally cause illness in the water birds, but they can sometimes be transmitted to other species, and they may become adapted to those other species. And so we know that there are many, many types of influenza A virus circulating in birds which don't cause any illness. From time to time they transmit to animals. Particularly interesting is this circuit here, where viruses can infect pigs or humans. And pigs are an interesting intermediate because they're able to be infected both by bird viruses and by human viruses. So they can be a place where these viruses get mixed up. And I'll explain about that in a minute. Now, we're most interested in this circuit, particularly the transmission from infected poultry, domestic poultry, like chickens, geese and other poultry, through to humans, and this transmission from swine, from pigs to humans. So typical zoonotic influenza viruses, that is viruses that go from animals to humans, that we worry about are H5N1 and H7N9, and I'll talk more about those. Now, again, to understand this, we need to look at the structure of the viruses themselves. And here's an electron micrograph of what they look like, and you can see that they have all these spikes on the surface. So this is a schematic of those spikes, and there are two main proteins that sit on the surface of the virus and are also then on the surface of a virus-infected cell in our respiratory tract. They're the hemagglutinin, HA, and the neuraminidase, NA. These are important not only because they sit on the surface so the immune system sees them, but also because the HA is used by the virus to get into cells, and then the NA is used for new viruses to escape from the cells. It cuts the bonds that enables the viruses then to emerge and go on to infect other people or other animals. These two proteins are also the basis of our typing, H1N1, H5N1, H7N1. These refer to... Now, the other really important aspect of the virus is separate pieces of genetic material, RNA. Now, these characteristics of the virus are critical. So first of all, the HA and the NA, because they are the main proteins on the surface of the virus and on infected cells, antibodies, we make antibodies against them. And so these are the targets of human immunity. And that means that immunity is actually a selective pressure for the emergence of variant H and N molecules. It's really Darwinian selection happening in our bodies all the time when we're infected by a virus. And mutation happens at a very high rate in influenza viruses because they lack a way of correcting mistakes. And so that means these viruses are changing all the time. And if we're infected with a virus, we'll have progressively a great mixture of them circulating in us until we cure the infection. So this is the main site of variation in viruses between different strains. Now, I mentioned that the genetic material is in 8 separate pieces, and that means that you can get shuffling of those different genetic pieces when two different viruses infect the same cell. So let me just say that in a little bit more detail here, because it's just so critical to how pandemic viruses arise. The first one I mentioned was mutation, so we have random mutation just across any of the genes of the
influenza virus. And if that's happening in somebody who has antibodies against the HA or the NA, then there will be selection, immune selection for a virus with an altered HA and NA. This is called antigenic drift. It just drifts along all the time, and it's the reason we need to update vaccines. This other process is called reassortment, and this happens if you have two viruses infecting the same cell. Then you have the chance to make new combinations of genetic material, and so you could have, as shown here, a new hemagglutinin mixed up with a different neuraminidase, and now you have a new virus. And that's what a pandemic virus looks like. It's capable of spreading between humans. So mutation causes progressive, and that's called antigenic drift, and this is the reason we need to update seasonal influenza vaccines quite frequently. And of the at least three, possibly all, of the last four pandemics, 57, 68, 2009, possibly also 1918, caused by reassortment, which caused this dramatic change, which is called antigenic shift instead of drift. And what you have in a pandemic usually is a new HA that's unfamiliar to the human population, sometimes a new NA and sometimes a mixture of different internal genes. We have various ways to treat influenza or to prevent it. Vaccines are the most important because they can prevent infection, and they work by inducing antibodies against the HA, and then that blocks the ability of the virus when it comes to the person to infect the respiratory tract. There are other cell-mediated responses which are not very well induced by current vaccines. Then there are antiviral drugs. There's a group of drugs called the adamantanes, but all these circulating influenza A viruses at the moment are resistant to these drugs, so they're not really of any use. But there are a group of drugs called the neuraminidase inhibitors, and these are highly effective if they're used very early in the infection. They're also very useful for treatment of extremely ill influenza patients. The famous ones are Tamiflu and Relenza. These are ones widely used in the world, especially Tamiflu, but there are new ones coming online in some countries. So the challenges of influenza are influenza viruses cause significant illness and deaths worldwide and have a very high economic impact because of effect on work. And these viruses are highly changeable, able to undergo mutation, and so we have emergence of drift variants. Co-infection with different flu viruses can lead to the production of pandemic viruses, and there's a huge reservoir of animals to supply material for the production of these new viruses. You can't predict the outcome of such reassortment. Vaccines have limitations because they have to be updated regularly, and they don't protect you against the next year's flu viruses unless the viruses aren't changing much. And drugs also can only be used very early in infection, so they're not optimal. So we have a continuing need to monitor seasonal and zoonotic influenza viruses in order to be able to try to update vaccines, to have suitable drugs, and to have public health preparedness in the event that a new influenza virus emerges. So let me talk about surveillance. The main surveillance network around the world, not the only one, but the main one, the largest one, the one involving the most countries, is the WHO, World Health Organization, Global Influenza Surveillance and Response System, which we call GISRIS. Not all that nice, but GISRIS, we know what it means. Now, this has been going now for a long time. It was established quite soon after WHO itself, which was formed in the late 1940s, and it monitors the evolution of influenza viruses infecting humans. Various recommendations, which I'll talk about. It serves as a global alert mechanism when new influenza viruses, potential pandemic influenza viruses emerge. It collaborates with the Animal Health World Organization to exchange information on human and animal viruses. This is increasingly important. And it has 6 peak laboratories, of which the one that I'm from is one. 5 of those are for human influenza, and 1 in the US is for animal influenza. So this network looks like this. It has, at its absolute heart, WHO national influenza centres, which are now numbering 141 in 111 countries. So more than half the countries of the world have at least one national influenza centre. So Indonesia has the WHO National Influenza Centre at NIHRD in Jakarta. And that's a very important component of this system because Indonesia, of course, has a very large population, and we know you have influenza here, and so the role that NIHRD plays is really critical in this process. So those centres theoretically cover more than the world's population. Then there are the 5 human collaborating centres: Atlanta at CDC, London, Beijing, Tokyo, and Melbourne. You'll see we're the only one in the southern hemisphere, so we have a special responsibility for south of the equator. And then there are some other laboratories that are involved, and we're all coordinated by WHO in Geneva. So the national influenza centres and the collaborating centres have different but complementary roles, and this is really critical to how the system works. The national influenza centres are selected by the Ministry of Health in their own country, and then WHO recognises them as a national influenza. They report what's happening to WHO regularly. They diagnose type or subtype influenza viruses, and then they send representative viruses or anything that's worrying for further analysis. And then the collaborating centres, like our own in Melbourne, we are designated by WHO to collaborate with WHO. WHO itself, we collaborate with WHO, and it's a great honour, of course. Our funding comes entirely from our own government. WHO doesn't give us money, but our own government enables us to do the work for WHO. We undertake detailed analysis of the viruses, which I'll talk about. We monitor for the emergence of new viruses. We monitor antiviral drug resistance. We report the data back to the centre that sent us the viruses. We isolate potential vaccine viruses, which we will then supply to manufacturers anywhere in the world if they're selected to be part of the next vaccine. We work with WHO to recommend which viruses should go in vaccines. And then we provide training, advice, kits, all sorts of things to national influenza centres as far as we can in response to need. And we also take research. You can see here how there are dots which are sending viruses to a WHO collaborating centre. And so the red ones here are countries or islands which regularly send viruses to our Melbourne centre. But you can see South America mostly sends viruses to US, and so it goes around the world. So there's quite good coverage. The main gaps at the moment are in Africa. We have very good coverage in Asia and the Pacific in general. So what happens when, for example, NIHRD sends a package of samples to a WHO collaborating centre, which it normally does to the US, to CDC? They, or we, if it's our centre, will ask, what influenza subtype or lineage is it? Does it differ from what's already known about what's circulating in its ability to be recognised by the immune system and in its genetic sequence, the sequence of the RNA genome? Is it detected by current diagnostic tests? If you had antibodies raised against a previous infection or a vaccine last year, would you be protected? Would your antibodies recognise that new virus? Is it sensitive to antiviral drugs? How far has this strain of virus spread in the world? And if it's a new strain, can we isolate one that could be useful for the vaccine manufacturers to produce a protective vaccine? So what that means in practice in our centre is that a national influenza centre will send us either a virus that's already been isolated by growing in cells or the original clinical specimen, sometimes both. We will do a couple of things. If we just get the clinical specimen, we can do a PCR straight onto that specimen and find out what the influenza virus is. And we can do a full genome sequence if we need to, if it's a good quality, fresh specimen, to determine the sequence of the virus that's in it. But we also attempt to isolate and grow the virus in cells. And this is a mammalian cell line, because then we have enough virus to do a much more detailed analysis, and this is really critical for our work. The first is to be able to look at the antigenic properties of the virus, its ability to be recognised by antibodies, and we use a couple of different assays for that. It's much easier for us to do gene sequencing, full genome sequencing. It's then possible for us also to do testing enzyme assays to look for sensitivity to the neuraminidase inhibitors, Tamiflu and Prolenza. And then we also look at the ability to be recognized by antibodies from people who have been vaccinated with last year's vaccine. With some of these specimens, we'll also inject them into eggs to try to grow the virus that way. And that's because for seasonal influenza vaccines around the world at the moment, the manufacturers are only allowed to make the vaccine from a virus that has not been into a mammalian cell line. If it's been into a mammalian cell line, it could be infected with mammalian viruses or other pathogens. If it's gone straight into an egg, then the risk is much, much lower because most chicken viruses will not be a problem for humans. So that's required by the regulators. And so for us to be able to provide vaccine candidates, we have to isolate the viruses directly into eggs under very clean conditions in regulated laboratories. So what we do is a really interesting mix of very old-fashioned technology. The hemagglutination inhibition assay, which is used for antigenic analysis, was developed in the 1940s. It's still the best assay we have. It's extraordinary. But then we do it in this fancy robot. So we've got this 1940s technology done in a 2013 robot to try to make it possible for us to do large-scale work. We do this old-fashioned methodology developed by an Australian, actually, for isolating the virus into eggs to make vaccine candidates. But then we do sequencing, pyrosequencing, whole genome sequencing, and now next-generation sequencing, deep sequencing. So using absolutely the latest technology to be able to understand genetic properties of these viruses. So it's a very interesting mixture of and show how they can continue to work together. Now, this is impossible to read, and this is a typical output of an antigenic analysis, a hemagglutination inhibition assay, and it takes a little bit of practice to learn how to read this and to understand it. But our collaborators at the University of Cambridge in England have developed this very nice way of mapping the data so that we can look at it in a simple to see and understand two-dimensional form. It's called antigenic cartography. And so this shows, just for PDM09 viruses, that the great majority, each dot represents a virus, are very similar to this red one in the middle, which is the vaccine strain, which is one of the viruses that emerged at the beginning of the pandemic in the US in 2009. There are just a few viruses different, and these mostly turn out to be artifacts. These are viruses which, when they're cultured in cells, have undergone changes that make them different from the ones that are circulating in humans. So because the recent viruses are so similar to this one, we haven't needed to change the vaccine. Now this shows the Yamagata lineage, and you can see there are two groups. Most of them are like this virus that's called B. Massachusetts 2 2012, belongs to a genetic group, clade 2, and that's the current vaccine virus for B. Yamagata, because most of the viruses we have look like that. There are still a few viruses which are similar to the previous vaccine strain called B. Wisconsin 1 2010. So this is a very easy way of seeing whether the viruses are changing or not, and then to know which ones to look at to understand better why they look different from the vaccine strain. This is really central in a collaborating centre. Now, the genetic analysis generates vast quantities of data that allows you to make a family tree of these viruses, a phylogenetic tree. And so what this shows is actually a small tree that I asked Yimo Deng in our centre to put together for this talk, because it shows a few Indonesian viruses which were sent last year from NIHRD to US CDC for further analysis. And I'm not sure whether the sequencing was done in Jakarta or in Atlanta, but nevertheless these are Indonesian viruses there that have the green asterisks. So this shows green viruses from Asia, including the Indonesian ones, blue ones from Australia and New Zealand, and the red one, which is there, that big one there, is the vaccine virus. And so that shows that most of the viruses genetically are in this clade 2, and that the Indonesian viruses are just very similar to the other B. Yamagata viruses that are circulating in Asia and Australia and New Zealand, as well as in North America and in Europe. And so that says here, Indonesian viruses are antigenically similar, also genetically similar to the viruses that are circulating in most of the world. And then we have a slightly different situation with the B Victoria viruses, where all of the Indonesian viruses are in one little clump here, highly similar to each other, highly similar to some viruses from Singapore, particularly maybe one here from Victoria, where our centre is. And you can see in this case they look like a separate little group that's managed to spread through a number of places in Indonesia as well as Singapore. Maybe it came from Singapore. But even though they look a bit different genetically, they're all antigenically similar to the other clade 1A viruses that are circulating in our region, and still very similar to the vaccine virus for that group of viruses. So we might see a group like that and think, well, it looks the same at the moment, but maybe in another year the descendants of those viruses will be different, so we'll keep an eye on that group. That's the sort of way that we work. Then the Cambridge group can put together tens of thousands of viruses into one tree, make this enormous thing, which is full of thousands and thousands of viruses. They're colour-coded. They show time here, different groups occurring in different months of the year here. And so we can, with putting all the data available to the collaborating centres together, we can get this large picture of what's happening around the world, because the national influenza centres each send in some viruses to contribute to this analysis. So why do we make a vaccine change from time to time? Well, we need to see several things for that to be justified. It's not something that one would do on a whim. One would not just do it because it feels like time for a change, because it costs the manufacturers a lot of money to change the vaccine viruses, and it's a lot of developmental work to make a new vaccine virus, and so therefore we don't want to increase the cost of vaccines by doing anything unnecessary. So we're looking for several things. We're looking for a marked change in the ability of the viruses to be recognised by antibodies compared with previous vaccines changes. We're looking for changes in the sequence of the hemagglutinin around that part of the molecule, and this is a molecular diagram of the hemagglutinin molecule, and the important positions are all here on the top, because that's part of the molecule that interacts with receptors on cells which are getting infected. It's also the part of the molecule that's recognised by antibodies, which are effective in protection. So we're looking for changes that would explain this, because we need to see some consistency there. We're looking for loss of antibodies from people who have been vaccinated with last year. See all of that, we're seeing strain, unless we've got a suitable, have to have a good virus that's been isolated in eggs, which represents the new strain with these properties. So we have to see all four of those things before we will recommend that there's a change in the composition of the vaccine. So this attempts to show all the changes that have been made in the three components of the vaccine, H1N1, H3N2B, since 2003 through to the most recent meeting that we had in Geneva in 2014, in February. And I've just colour-coded them. We meet twice a year. In February, we make a recommendation for the next Northern Hemisphere vaccine. In September, we make a recommendation for the next Southern Hemisphere vaccine. And you can see that for the H1N1s, we hardly ever change. They are much more stable than the other viruses. And in fact, I think this is really amazing that the new pandemic virus, which emerged in 2009, has still not undergone antigenic drift. We see a lot of genetic changes, but there's still no antigenic drift. So it must happen eventually, because just about the whole world has been infected. Most people will have antibodies against this virus, and you would think that would create the selective pressure for a new version of this virus to emerge. It hasn't happened yet. So we keep watching and wondering when that will be. The H3N2 viruses, you can see from the change of colours that they are much more changeable, and we have to keep a very close eye on them because they do change quite frequently. And then the B viruses also change quite frequently, and we're often switching backwards and forwards between a Victoria lineage virus and a Yamagata lineage virus as they become more or less predominant in the world. So now a bit about zoonotic influenza viruses. Why do we worry, and which ones do we worry about? Well, this is really a story about the world we live in today. And The first reason to worry, zoonotic viruses are ones that go from animals to humans and can cause severe disease and may cause a pandemic. We have intense animal husbandry and cross-border trade of animals throughout the world today. Of course, this is particularly intense in Asia and Africa, and many people are in live bird markets, for example, are mixing up different species, and farmers are bringing in their poultry from many different districts into a single market. So there's tremendous opportunity for infection of uninfected poultry and for mixing to create new combinations. And so as the world gets fuller of people, it also gets fuller of poultry and pigs and all of these other animals which are a worry for us for transmission of zoonotic viruses. China has about 12 billion poultry, so more than 1 billion people, but 12 billion poultry. So it's a very full world from an influenza point of view. High population density. Now, this is a map from a few years ago, but we know it's still true today and that some parts of the world are getting fuller and fuller. When I went to school, I was told that Java was the most densely populated region of the Earth. Is that still true today? Yeah, so many people in a small area. But of course, we can also see, so that's clear here, but we can also see other parts of the world where there's incredible density of population and enormous cities which are then surrounded by poultry farms and animal farms. So this is a big challenge if you think about how do you stop transmission of viruses. It's very challenging when we have a crowded world. And I should just point out, there aren't very many people in Australia, but in fact we're the second least populated country in the world. The other is the least populated area of the world is Mongolia. But in Australia, there are very few people in this desert in the middle, but we have some quite intense foci of population in our big cities, Melbourne, Sydney, and Brisbane in particular, Perth over here on the west. So we do have cities where there's a great deal of spread of influenza. And then this will be hard to see when the light's on, but I think you'll get the idea. It's the intensity of airline traffic throughout the world. Very impressive. It takes only a few hours for a new virus to get from one side of the world to the other, particularly if you're somewhere on this line, but also, of course, through Singapore. This is probably Jakarta here, I'm not sure, and even down to our relatively unpopulated Australia. So we spread our viruses very quickly. Where will the next pandemic come from? Well, the fact is we don't know. It's unpredictable. We don't know when it will happen, but there are certain viruses that are very worrying. And so avian H5N1 is a possibility, H7N9 in China. Other H7 and H9 viruses are prime candidates to become pandemic. That would happen by further adaptation of the existing viruses to enable them to become spread easily from person to person. Or perhaps that will be achieved by reassorting, by mixing up with the genome of another influenza virus that already has those properties. It could be that the pandemic virus, which emerged in 2009, will undergo further reassortment, for example in a pig, to create a worse pandemic virus. That's quite possible because this virus, which we have got from pigs in the first place, it first emerged in North America in 2009 or late 2008 and transmitted to humans from pigs, this virus very easily goes from humans back to pigs. And so we are a risk for pigs, and then in the pigs you've got the opportunity for mixing up with other swine flu or with other avian viruses to create a new version of that virus. And there are many swine viruses in Asia, USA, Australia, and some of them occasionally infect humans. But actually, in the end, we don't know. And just as with every other pandemic, it'll probably be something we weren't expecting. And so we have to watch and try to be alert. Now, I've listed here the major viruses we're concerned about. I won't go into detail here, but you can see that most of them are infecting humans from poultry, and they're quite widespread in the world. There are a couple which are particularly worrying, and I want to say a few more words about now. Both of them can cause very severe disease in humans. Highly pathogenic H5N1 in birds and low pathogenic H7N9 in poultry, possibly in other birds as well. So H5N1, and I have a feeling there'll be people in this room who know a lot more about this than I do, because it is a virus that's circulating in poultry in Indonesia, and Indonesia has been one of the main centres to be looking at the activities of this virus. So these are viruses that mostly cause mild illness in birds, but this particular highly pathogenic H5N1 virus, it causes severe, rapid onset of disease in poultry, and farmers can see their flocks basically all drop dead in 24 or 48 hours. So it has a very big economic impact. From a public health point of view, that has the advantage that you can see where that virus is. So when you see the poultry dying, you can, if it's recognised and understood, and there's a good system of interaction between the farmers and the local authorities, then there's the opportunity to clean up those flocks and try to reduce the further transmission. But of course, there are many reasons that this is very challenging. But at least you know where the virus is. It's spread very easily from contaminated birds. The spread around the world is mostly through trade in infected poultry, partly by migratory birds, but mainly really through human activities. It's not all that easy for humans to catch this virus. When it happens, it seems to be usually with very close contact, so the virus can get right down into the lungs where it will find the receptors it needs to infect. But when it does infect humans, it looks as if the mortality is still extremely high. About 60% of known cases reported to WHO die of the virus. But luckily, mostly this is not passed from one person to another, so it's not a pandemic virus yet. You can see that the cases, this is recent reporting to WHO, is highly seasonal, so it's actually the same as the seasonal human influenza virus season. It looks as if the numbers of cases are dropping. I don't know whether it's really dropping, whether this is just because fewer cases are being reported, but it's very encouraging that the numbers are dropping. The spread of the virus hasn't declined, but cases in humans appear to have declined. Now, this is a very complex genetic tree, family tree for H5N1 viruses put together last year by a working group that I'm a member of, and which has just been published quite recently. And what it shows, you can't possibly read it, and in fact, if you look at the journal article, you can't read it either. It's been so badly reproduced. But anyway, what it shows is that there are now many, many different groups, genetic groups of these H5N1 viruses. There have been a whole lot of genetic groups which have disappeared, so-called clades, but there are several, 1, 2, and 7, and these are all group 2 here, and then group 1 here, and then group 7 here. These all continue to undergo genetic change, and some of these samples have been analysed in animal labs or in WHO collaborating centres, and we know that they're antigenically different. That means you can't use one vaccine for all of these different viruses. You need in Indonesia a different influenza, H5N1 vaccine, from what might be needed in Egypt or Israel or India and Bangladesh, for example. So this is a huge challenge. If we want to work globally to have vaccines to protect against these viruses, each country has to decide what it's going to do, and it also almost means each country has to make their own vaccine. This is a very big challenge, particularly for countries which don't have vaccine production facilities, which of course is not with Indonesia. You have an excellent facility production system here. So the threats are that H5N1 viruses continue to circulate across several continents of the world. Nothing has stopped that yet, and they are very well established in Bangladesh, China, Egypt, Indonesia, and Vietnam. They've diversified, so they're no longer covered by one vaccine. They still cause human deaths at very high mortality, and there was some very interesting, very controversial work published the year before last, which showed that it only will take a few more changes to these viruses to enable them to transmit easily between mammals, so potentially between humans. That doesn't mean those changes will occur, because there may be reasons the viruses can't undergo that evolution, but it looks like these are viruses which are just teetering on the brink of being able to spread easily between humans. Now, H7N9 emerged last year in China. The first known case started in late February. It was picked up very quickly in China because they have a good surveillance system for pneumonia of unknown origin. And so clinicians in Shanghai recognised they had 3 family members with severe pneumonia. They isolated the viruses. A lot of work was done, including at the WHO Collaborating Centre in Beijing. And as soon as China was certain it had a new virus, it put the sequences into public databases available internationally, and they announced this new virus to the World Health Organization. And then we all knew about it, and everybody could start working to be prepared in case it was a problem. And so I think that's a very big change from China a few years ago when SARS came out and they tried to keep it a secret. So China has done extremely well here, and it's meant we now understand a lot about these viruses, which we would not have otherwise done. Now, these viruses are a reassortment. They're a mixture of genes from several different bird species, and just by chance they turn out to infect humans, and they caused a big spike of infections, quite a number of whom died in pale blue there, just through this period, March, April, May last year. Then it got very quiet for a while, just a few infections. We hoped it had gone away, but of course when the season changed, then they came back again, and there's been a much worse season this year, which is still continuing now. So these viruses are continuing to circulate and occasionally infecting humans, and because, of course, the population is very large and very dense in China, there are a lot of cases. They've now been isolated in humans through this enormous area of China, and there have been imported cases into Taiwan and into Malaysia. Not yet beyond there, but in a sense it's just a matter of time. We expect that it will happen. So it was recognised very early that the people who had these severe infections, they were mostly older men in big centres like Shanghai. Most of them had a connection with live poultry markets. Men who are retired tend to do the shopping. So they're going each day to the live poultry market buying chicken to bring home. That sort of thing. And so very quickly, Shanghai, just a few days after, 31st of March was the day that China announced to the world that they'd discovered this new virus. Shanghai closed all of its wholesale and retail markets on the 6th of April, culled lots of chickens. I took that photo when I was on a WHO mission there in April, an empty wholesale market, compared with the sort of thing that would have been there before. And so there's actually some quite nice data in a paper here which shows that in different regions of China where there were a lot of cases, the cases fell away once they had closed the live bird markets. So the combination of closing the live bird markets and a change of season meant that that virus appeared to vanish. But of course it's come back again. The markets are open again in most places, and so the problem continues. Now, I won't go into this, but there's very important features of this virus. The fact that the sequences were made available internationally meant that groups could very quickly do detailed analyses of these sequences, and they've recognised that the viruses have a whole series of features which are listed here, which mean that the virus is better adapted to infect humans. Can't yet transmit easily between humans, but it has a number of features that enables it to infect humans. So it's now on the top of the list for pandemic concern. And so I won't go through this, but you can see the pandemic risk is difficult to assess, but we think it's greater than for highly pathogenic avian H5N1. So WHO has done a lot to prepare for a potential pandemic, and it shows really the advantage of this global cooperation, if you like. It's provided technical guidance, it's provided diagnostic tests, and the labs have produced kits that can go all over the world so that labs can be ready to diagnose these cases. If you have a suspect H7N9 case in Indonesia, then NIHRD probably has the kits to be able to diagnose that quickly. A lot of work has been done to go through the early stages of vaccine development so that if this virus starts to spread between humans, it won't take as long as usual to make a new vaccine. And so these viruses are causes for continuing concern, and that's really because elimination of the bird reservoirs is probably impossible. it is certainly impossible now for H5N1. It's spread so far in the world. H7N9, well, we don't really know the origin of it. We don't know where it's come from, and so it's also probably very difficult now to get rid of it. But we can reduce the risk through controlling breeding and trade and live bird markets and other practices that favour the generation of these sorts of viruses and favour spread between birds, other animals and humans. And we need to have this continuing surveillance and analysis of viruses so that we can realise quickly if there's a change in the viruses that mean the risk is now higher. Or the risk is lower and we needn't worry as much. But we're more worried, of course, about the risk being higher. And so we need to be able to evaluate whether our interventions are working and know whether we need to start making vaccine, for example. We also need better vaccines. We need vaccines that produced this year could protect us against something we don't know about yet. And so our own group in Melbourne and many other groups around the world are doing a lot of work towards that. I won't have time to tell you about that, so I'm just going to flip through these research slides because there are way too many of them. And
can we wrap up in 5 minutes?
5 minutes? I could stop in 1 minute if you like.
No,
I think that would be better.
I interrupt you in the middle, so I can spare you 5 minutes.
Yeah. So, no, that's fine. I thought I would finish at 10:30. And it's almost 10:30. So I wanted to just say, I've told you about seasonal pandemic and zoonotic flu viruses, global surveillance and zoonotic viruses. I haven't had time. I didn't think I'd have time. Another time maybe, tell you about some of the research we're doing. And I just want to finish one slide, which really is about the prospects for control of influenza. A lot on this slide, but just the same, I'm trying to encapsulate here what I think is the situation now. We live in a very different world today, and I've emphasised the risks of that world, but there are also many benefits we have, and so it's a mixed picture. So today we have much better surveillance for influenza in many countries. We have rapid specific diagnostic tests, PCR-based tests and rapid tests. We have antiviral drugs that work if they're given soon enough. We have strain-specific vaccines, not the best vaccines, but a lot better than nothing. And we have a much better understanding about how these viruses work. We know what to look for much better than we used to. And in many countries, not all, we have better ways of keeping domestic animals separate from other domestic animals to reduce the risk of transmission of viruses between them. And I'd say one of the most important things we have today is rapid global communication systems. If you go back to 1957 and 1968, it took a while to realise that new pandemic viruses were emerging in China and Hong Kong. But nowadays, it doesn't take very long at all. But there are still a lot of challenges. Many countries don't measure the impact of influenza on health and the economy, so they're not sure whether they should be bothering to spend money on vaccines because they don't know what the situation is in their own country. So it's important to try, where possible, to get the data to know what decision to make. Of course, countries have many priorities for health spending, and we're about to hear about another one of them. We know that throughout the world there's unequal access to vaccines, drugs, and other mechanisms for control, and that's something we all want to see overcome. Traditional farming and trade practices, which I've emphasised, are still prevalent in many parts of the world and will continue to be so for a long time. But connectedness of people, animals and products is ever increasing. We don't have the best vaccines that would save us from the next pandemic. We have to make a vaccine when the pandemic arrives. And we know that these are viruses that are very good at evading immunity and evading antiviral drugs. So my feeling is that the response to this, the ultimate goal, has to be, I mean, I'm a scientist. I would love to think that science has all the answers, and I know science has some of the answers. But it's also true that it depends on social, political, economic activities that will drive the kind of global cooperation that ensures we reduce the impact of this and other viruses on human populations. So thank you very much for your attention, and I look forward to talking more later.
Thank you so much for the wonderful presentations and very broad and comprehensive. We might save 1 or 2 slides on the current research. Yeah. So you can all. Would you like to drink first, or? Yeah, I would like to tell the audience that because I would like also to tell you that many of these are researchers, so it might be very relevant for that. So I have to apologize for stopping off at the time, but considering the audience, I think it would be good to motivate the younger researchers. Yeah.
I'll tell you about one project which is a very important part of what we're doing in our centre. Comes soon. And just to start with, our centre and part of the University of Melbourne, part of the Royal Melbourne Hospital, just moved into the Peter Doherty Institute for Infection and Immunity, and it really is as beautiful as this. And our labs are just here. This is the tea room here, and this is us in here. And it's really a wonderful place. So I really hope you have the opportunity to visit us there. And it's a centre now for collaborative research and education and diagnostic and reference laboratory work as well. And it's surrounded by other great institutes like the Walter and Eliza Hall Institute, the Florey Institute for Neuroscience and Mental Health, a big new cancer comprehensive centre being built next door. So it's a real hub in Australia for this type of research. So I hope you can visit. And so one of the areas of special interest to us is antiviral drug resistance in influenza viruses. And this is work that then grows directly out of our surveillance activities, which I think is a good model for reference labs. Now, antiviral drug resistance in seasonal influenza viruses is pretty interesting. I've told you about Tamiflu and Relenza. They're actually synthetic drugs. This is Tamiflu. And they're active against both type A and B viruses. They work by blocking neuraminidase acting with its blocking the active site so that neuraminidase can't release new viruses from infected cells. Sometimes viruses will acquire mutations which mean that they can resist the drugs. But usually when that happens, the virus also loses the ability to be an infectious virus, because by interfering with that active site of the neuraminidase can no longer do its job properly. And so mostly when a drug-resistant virus emerges, it can only really succeed in a patient which is treated with the drug. Because if it's out in the general community and infects the general population, the other influenza viruses are much more ecologically fit in that environment, and so the new drug-resistant virus is very unlikely to succeed. It's really an ecological argument. But something special happened in 2007. It was picked up in early 2008 when Norway realised that most of the H1N1 viruses it had circulating in Norway were resistant to Tamiflu, and they had a very particular mutation, histidine to tyrosine, at position 275 of the neuraminidase gene. And the amazing thing was that then we all started looking and we realised these viruses were spreading all around the world. It took 1 year for the H1N1 viruses around the world to go from being 100% sensitive to being 100% resistant to the main antiviral drug we were using, Tamiflu. So that was an interesting situation, but then we had the pandemic. In 2009, just a year after these all became resistant, the pandemic came along, and it was a new version of H1N1. It was sensitive to Tamiflu, and it displaced the old H1N1 from the ecology of influenza viruses infecting humans. So we were very fortunate that while it's not good to have a pandemic, in this case it actually pushed out a virus which was causing us a lot of problems. Now, a very interesting thing was recognised by a group in the US, that the reason this H275Y mutation had become established in H1N1 viruses was because other compensatory mutations had already occurred, which meant that the mutation didn't interfere with the fitness of the resistant viruses. And my collaborator at the centre and I, Aaron Hurt is the main person who does this work in our centre, he and I wrote this little article for Nature Medicine, and we drew this picture to try to illustrate it. And it shows here how the normal situation is if you treat a patient with Tamiflu, it might produce some resistant viruses, but when they're transmitted to the next person, they don't succeed and they don't go on. But if you have some compensatory mutations, as shown here, then you might start to have a virus which is actually able to spread between people who are not being treated with the drug. And what happened in 2007-8 was that all the viruses became like this, and so all people around the world who had this infection had a drug-resistant virus. So in 2009, we now had a pandemic. We had a new virus which was Tamiflu sensitive, but we recognised in 2011 in Australia that we had a large cluster of resistant pandemic viruses. And this was the largest cluster reported up to that time, and it remains the largest cluster reported, of Tamiflu-resistant viruses that could circulate to people who were not being treated with the drug. It was centred around the city north of Sydney on the coast of Australia. We have most of the cases there. We have one case 4,000 kilometres away in Perth, so probably somebody took a holiday. And so this was very interesting, and we've been doing a lot of work to understand why this could occur. We were again very, very lucky that even though there were a lot of these cases, when the end of the season came in Australia, that virus died out. We've watched to see whether it would emerge anywhere in the world, and we've been very fortunate that it hasn't. But we've been doing research, particularly Aaron Hurt with collaborators has been doing research to see if there are permissive mutations that enabled that cluster to happen and which might increase the risk of it happening again in the future. And Aaron has discovered 2 mutations. This shows the frequency of a number of different mutations over time from 2009 up until late 2013. And it shows these 2 mutations which came up in 2010 and are now present in about 95% of all H1N1 PDM09 viruses. And it turns out when we do a whole series of experiments to examine the fitness of these viruses, that these mutations make these viruses more able to acquire the Tamiflu resistance mutation and still be infectious viruses. I hope that's clear. So it looks as if, one of the ways this has been done is by using the ferret animal model of flu infection and using mixtures of viruses with and without these mutations to look at which one wins. And so you can then sample the ferret every day and look at which viruses are there and show that if they have one or more of these mutations, then the drug-resistant one is able to survive. So that's really just a small snapshot and really now raises the question. We've gone from a situation where in 2009-10, any virus which acquired the H275Y mutation did not continue to spread, to 2011-12, where it looks as if we probably had a situation of moderate viral fitness that allowed that Newcastle cluster to happen. And now we're wondering what further changes are needed for these viruses to become completely resistant and spread around the world. So a very important activity that's a mixture of surveillance and research in our centre is monitoring viruses for further changes and seeing whether this event is likely to occur. So that's all I wanted to say about that. I hope that's interesting.
Very interesting. Thank you so much. Again, have an applause for Professor Kelso.